I'm trying this new system for recording that I'm hoping will be better than what I've done in the past. With usually I can like, you know, do different things with it, put information, like if I need to like pause and like put some clarification into it. Hopefully I should be able to do that. So, antigens and antibodies in blood banking. So this is the core of everything we're doing in blood banking. We're doing antigens and antibodies on the red cells and in the plasma. So we're going to talk about characteristics of red cell antigens, characteristics of IgM and IgG antibodies, where we talk about factors affecting sensitization versus agglutination, and we're going to talk about reading and grading reactions. You guys are also going to be taking concurrently with this class basic immune concepts. This is another reason why we're moving this in third semester, because it'd be really helpful if you had basic immune concepts before this lecture, because we're going to talk about antibodies. And so what happens these first couple weeks, we're going to have a lot of like, I'm going to talk, they're going to talk, I'm going to talk, and we're going to be kind of like learning at the same time about these topics. In a way, it's great. You have two of us kind of explaining different things, but in a way, it's also frustrating because we're bouncing back and forth a lot. So, red cell antigens. This is a red cell. This is the inside of a red cell. This is the exterior of the red cell. So this is the cell wall of a red cell membrane. It's a biphosphate lipid layer is what it's called. This you're going to cover in hematology today and next week, probably. Is another reason. We're juggling too many things all at once. But this layer, this is the outside, this is the inside. So what happens with these antigens is they are somehow on the outside. They're either attached to the outside of the biphosphate layer. They go through the wall, through the membrane. Sometimes they start on the inside and go through, and sometimes they start in the middle and go through. So, but they show up on the outside. That's what we're worried about. We are worried about red cells. So the red cell membrane is what you're looking at, that biphospholipid layer, which you'll talk more about. But what we're looking for are things on the outside Those are your antigens. They are foreign to the recipient. Now, your red cell membranes are, your red cells aren't, right? So let's say this is what you got on yours, but then I'm given donor blood, and on the donor blood, instead of a triangle, circle, square, I've got a circle, a rectangle, and we'll say this is a hexagon. Okay? Those are different than what mine are. The circles might be the same in the different areas, but the rectangle and the hexagon are going to be different than my triangle and square. So foreign to the recipient. Large and chemically complex proteins and polysaccharides. Oftentimes they're made of proteins and polysaccharides, and they're large and chemically complex. If they are small and chemically simple, they're not going to be as big of a problem as the larger ones. We'll talk more about that in a little bit here. Autologous antigens, auto means self. So when we're talking about anything autologous, it's on your own cells. So these are my autologous antigens. The homologous or allogenic, present on the cells of other humans, these are the homologous ones, the ones I'm given from somebody else. This is the picture. Nope, this is not the picture I handed out. This picture will be coming later when we talk more about some of these at a later point. So you're going to learn about all these different cells later. Right now, what I want you to get from this picture is to understand that these are all different types of antigens that can be on the outside of the red cells. There's over 300 groups of antigens that we're going to see on the outside of these cells. I'm not making you learn all 300. I'm making you learn about 25 groups, 20 groups? Nah, not even that many groups. I'm probably going to make you learn about 15 groups, okay? You can see, like, these P's are attached to the outer membrane. These guys are going through the membrane. See this one here? This one wraps in and out of the membrane over and over and over. So it depends on which antigens we're looking at, how they're going to be detected, how much trouble they're going to create, and how to make them stronger for us to test or weaker if they're hiding something.
Like in the last picture, it had glycoproteins and glycolipids. antibodies, or is it just one of them?
So if you notice, like, some of these are going to be, how do I explain this? Don't go there yet. You aren't quite there yet. I'm trying to think through how to do this. You're not quite there yet. Great question. Know that they're going to be proteins and carbohydrates mostly. So that's what when you're looking at the polysaccharides, you're looking at sugars, right? Versus proteins. So depending on the antigens, we go through them the groups wise, you'll find out what their main structure is going to be when you're learning through them. Okay, I think that's all we'll talk about there for right now, because we will get into all those closer, one by one by one, like we did in your analysis. So, factors that affect antibody stimulating by a red cell antigen. So, how am I going to get my antibodies to say, whoa, my body needs to make, my body needs to say, whoa, I need to make an antibody to this. So we're going to look at the size and complexity of the antigen, the degree of foreignness, the dose administered, nutritional status, the age of the recipient, and then immunocompetence of the recipient. I'll go back for it. Okay, and you go back to this one. I thought I had individuals on this. So, let's talk about each of these groups together. Size and complexity of the antigen. The bigger it is, the more complex it is, the more likely you are to develop a response to it. Okay? If it's something kind of small and simple, your body's not probably going to recognize it as something different, because you got to think about all the building blocks we have. If it's just a small, some of those pieces, it's going to look like self. The bigger it is, the more complex it is, the more like self it's not going to look like. That goes to degree of foreignness. The more likely it is to your antigens, the less likely you are to develop antibodies. If it's not like your antigens, it's more likely to develop antibodies. You guys all know about ABO so far, right? Everybody kind of has a general understanding of A and B blood types and O blood types. So if I'm an A and I'm given A blood, I should not develop an antibody to an A, because it's just like me. You're going to learn it's not just like you. There's differences within the A, but we'll get there later. But if I get B blood and I'm an A blood, whoa, my body's like, this is not me at all. I need to do something about this, so I'm going to develop an antibody to it. The dose administered. If I start giving a unit of blood to a patient and the patient starts reacting, first thing we do is stop that unit. If I go, it's not that bad, and keep giving it, the more I give, the more response it's going to be. Some things we can give one dose, like 1 unit of blood, and they're going to develop an antibody. D, RH, the RH factor. If I give a unit of D, 1 unit, and you're likely to develop antibodies against that unit. Then we have other antigens. For example, let me think of a good one here. Oh, that one will too sometimes. I'm going to say S. If I give you a dose of S, it might take 6, 7 doses of S before you're going to develop antibodies. I might be able to give you 6 units, 8 units, 20 units before you're going to actually develop an anti-S antibody. Okay? Which again, that means nothing to you right now, but it will. So some little bits are going to create a response. Some, the more you get, the more response you're going to get. Nutritional status of the recipient. Poor diets do not allow good immune response. Think about that's with anything, right? Like when you're in the middle of midterms or finals, how many people here are eating really, really healthy, good foods versus grabbing a pizza? How much more likely are you to get sick during those times? You're not eating well, you're not sleeping, you're not taking care of yourself, right? If you're more likely to get sick, it means your immune system's not working. That is where the immune competence, I'm going to jump down here, of the recipient. If your immune system is poor, you are less likely to develop antibodies against mismatched blood. This is one place where a poor immune system is actually good for you. I'm not going to worry as much about it. So if you have somebody that's getting cancer treatments and getting a lot of blood, you're less likely to develop an antibody response to one of these bloods because your immune system's not very good. It's not reacting very well. And then age, very, very young and very, very old just are not good antibody makers. And by very, very young, we're talking, 6 months to a year. They're not going to be very good at it. It takes 6 months or so in the body before you really start developing good antibodies. And once you get a little bit older, your immune system starts falling out also. One way to think about this, when you think about antibody stimulation, think about, let's say I go to the mall. I go over to East Town Mall. Come home, talking to my husband. What am I going to tell him about what I saw at the mall? Am I going to remember anybody I saw? Right? I mean, you see hundreds of people at the mall, right? They're just like me, not very complex. They look like me, they dress like me. Not foreign. There's a lot of them, but they're all alike, so it doesn't matter. Not going to make a response, right? Let's say when I was at East Town Mall, though, there was a clown running around. Okay? Big red wig, makeup, nose, the big feet, He's going to catch my attention. I'm going to go home and say, there was this clown at the mall. Literal clown. I'm not, talking about the clown that cut in front of me in line, right? They're talking about the clown in the mall. If I happen to be in the same area the mall as him. Right? The mall's big. I may never have seen that clown. I may not have even had a response to it. But now, let's put 100 clowns at the mall. There's a really good chance I'm going to see more than one, right? Even if I'm at, I don't even know what's at this mall anymore. Even if I'm at one end and the clowns are spread throughout, I'm likely going to remember that there were clowns at the mall today. It was crazy. There were clowns at the mall. I'm going to tell my husband, but I'm going to tell you guys aren't going to believe this. I was at the mall yesterday, there were clowns everywhere, right? So now I have a bigger response because there were so many of them. They were different, and there were so many. Now let's put them on stilts. There's no way I'm missing 100 clowns walking around East Town Mall on stilts, right? I'm going to be telling you guys, I'm going to be telling Bob, Trent, and Kara, I'm going to be telling Marcy, I'm going to be telling the other teachers up there. I'm like, it was crazy. It was pandemonium at the mall. All these clowns running around on stilts. Because it made a bigger reaction for me. Versus any other day at the mall where I'm going to see 100 different people and not remember a single one of them. Make sense? That's kind of how we're asking our body to respond. If I get one red cell in my body that has Duffy, and I don't have Duffy, so it's not normal, the odds of my immune system, my B antibodies, seeing it before it gets destroyed, slim. Okay? If I get a bunch of them, my body's going to react. If it's an antibody I don't have, or an antigen I don't have, my body's going to react even more. Same idea. Kind of make sense? Okay, just some ways to remember that. Okay, so the antibodies we're talking about in blood banking, IgG and IgM. Just by looking, what's different? Size. Yeah, that's going to be the obvious one. Anybody know any of my antibodies yet? These are my reacting sites. What's something else you notice? Which one has more reaction sites?
IgM.
IgM. It's bigger, more binding sites. Okay. This is the picture that I handed out to everybody. This is like an antibody response, a very simple picture. I can't remember if this is in your blood bank book or your basic immune concepts book. It might be in basic immune concepts. I can't remember where I stole it from. The reason I like it though is it shows what's going on with multiple exposures. So you can see here, this is your first exposure. This can be to an antibody, like we're already talking about in blood bank. This can be to hepatitis B vaccine. This could be to an actual, it could be to an antibody, it could be to a bug, it could be to a vaccine, doesn't matter. What happens is these naive B cells are going to activate, make plasma cells, but then the numbers are going to kind of go down. And the plasma cells are going to hang out in the bone marrow. So there's this little bit of a response. IgMs are going to be the first ones to the site. IgMs are going to be the first to react. They're going to attack. The numbers are going to go down, but then you got a second exposure. So your second Hep B vaccine. You get exposed to a virus. You get exposed to a bacteria. You get exposed to an antibody or an antigen on the outside of a cell wall. The memory B cells don't need to be made. You don't have to like introduce it and activate your cells. They're going to be still being hanging out there. They're going to double. We're going to get more plasma cells, making a lot more IgG because it already went through that process. Your immune response is going to be quite a bit higher before it slows down. And you can see when it slows down at a level higher than its last down level. So this is what's happening in the body. Every time you get exposed to something, you get a bigger and a bigger and a bigger response. Hepatitis B, for example, that's why you need all 3 vaccines in a series. They have found that the hepatitis B vaccine series, the best one is at time 0, 1 month, and 6 months. That's how we get the highest response. Now, once you're done with that vaccine series, you go into the hospital and you get a needle stick, your body's going to be like, boom, on that, right? We want that to be a fast response where our body doesn't even have to think about responding if we get exposed to hepatitis B in the medical field. We want that response way, way up there, and that's why we give the 3. So each response is going to be slightly bigger than the previous response. So some important characteristics. IgM is going to be the first antibody formed. If you look back here, IgMs get formed first. They're a little more general, not as specific. They're going to come out first. They're going to be large, over 5 times larger than the IgG. If you look at this, this is one subunit, one Ig subunit. This has 5. There's 1, 2, 3, 4, 5. Sorry, kind of blurs there. 5 is a 5 subunit structure. So when I get back to here, so it's huge. Number of binding sites, there were 10. Versus IgG, second response, long term, it's much smaller, and there's only 2 binding sites. Complement activation is really good with IgM. If you don't know what complement is, you'll get there. Just kind of know that when we talk about complement activation throughout. IgG, if conditions are right, it could. Optimum temperature, 4 to 30 degrees Celsius. Cold. It likes refrigerators, it likes room temp. IgG likes body temp. Serum concentration, IgM is fairly low. IgG is fairly high. Placental transfer. IgM doesn't cross the placenta, it's too big. IgG does. This gets really important when we start talking about hemolytic disease in the newborn and Rh immunoglobulin. Clinical significance, usually IgM doesn't have much clinical significance because it's too big to cross the placenta and it likes it so much colder, where is it cold, that's not going to react much in body temperature, it's just going to be hanging out. The exception is your ABOs. Serum concentration of IgG is high, it does cross the placenta because it's so small, and it can cause hemolytic disease in the newborn and hemolytic transfusion reactions, so the clinical significance is a lot higher. Ways to think about this. IgG, T-Rex, okay? They liked it hot, right? It was hot on the world and on the planet at that time. They got tiny little arms, okay? If you threw like a huge ball to me, this is as far as I can grab. If it's too big, I'm not gonna be able to grab it and hang on to it, right? And I'm definitely not gonna be able to grab 2 because they're gonna be knocking each other off. So they're warm, they're small little arms. IgM, think of a Yeti. Big, they're cold. Big arms, big legs, they can grab things. They're all far enough, both arms, both legs, and my mouth, right? They can hold 5 things at once because they're so big. Of course, they wouldn't be falling on the ground, but you know what I'm saying, right? They're cold, they're from the mountains, they're from the snowy areas, right? So IgMs, you're gonna think Yetis. IgGs, T-Rexes. Lost my thing, there it is. Kinds of antibodies we're gonna find. Naturally occurring, formed without red cell exposure. They happen because of exposure to similar molecules, like in bacteria. IgMs in particular are going to be naturally occurring. Most common IgMs naturally occurring are going to be your ABOs. I know as a type A person, when I do my reverse type, I have anti-B in my plasma. Okay? Where did that get there? Did I get B blood at some point? No. My first 6 months of being born, I was exposed to normal bacteria in the environment, and the ones that look like anti-B, or look like B antigens, were so different from myself, my body produced antibodies to it. The ones that look like A antigen, my body didn't produce antibodies to because it looked like me. Immunoglobulins formed as a result of stimulus by red cell antigen. Blood transfusions and pregnancies, so we're going to see most of these. These are going to be IgGs, your immunoglobulins. Autoantibodies, antibodies to your own antigens. This is an abnormal condition. Frequently results in vivo cell destruction. So I'm destroying my own cells in my body because I have produced autoantibodies, antibodies against myself. These can be IgG or IgM. We'll talk a lot about some of those farther down the line. Alloantibodies, also known as unexpected antibodies, irregular or atypical antibodies. Alloantibodies, produced in response to foreign antigens. These may be IgG or IgMs. So these are produced because I'm exposed to blood. Remember I told you there's over 300 blood groups? And sometimes we have the A form, and sometimes we have the B form, and sometimes we don't have any of them. If I get ones with the other kinds of form, those are all going to be potential problems. That's what I'm talking about here, is I can develop antibodies to any of those antigens on any of those red cells. Agglutinin, capable of causing agglutination in vitro. So now we're talking in a tube. An agglutinin can cause agglutination in a tube with the correct corresponding antigen. So if I put anti-A into a tube with cells with A antigen, it's going to agglutinate. If I put anti-B in a tube with cells with A antigen, it's not going to agglutinate. I don't have the corresponding antigen. Oops, what did I just do? There, antigen antibody. IgM antibodies are really good agglutinins. They're big. They can hold lots of things, right? IgM antibodies, not very good. My IgM, I'm going to be able to grab one red cell and just hang on to the one and not cause an agglutination, right? It's just going to hang on. That's called sensitization. I'm going to hang on to it. I'm going to sensitize it. I can't do much else because this arm's not going to be able to reach anything else. Versus IgM, I got one here and one there, and now look, I got an agglutination because I got two cells. Hemolyzed causes hemolysis when reacting with the corresponding antigen. Oftentimes, this will be due to complement activation, or if we remember, IgM is really good at complement activation. Anything that's a hemolysis is considered to be very clinically significant. This is where we have problems in vivo sometimes. It can also be in vitro, but this is where it causes big problems in vivo if I have a hemolysin that's destroying my red cells. Maybe an IgM or an IgG. Isoagglutinins. This term is sometimes ones we see to describe those naturally occurring ABO antibodies. So like I said, I'm A blood, so I have anti-B antibodies. It's an isoagglutinin. It's naturally occurring. We pretty much talk about them, and particularly about the A, B, and O blood groups. Cold agglutinins are going to be ones whose optimal temperature is going to be less than 30 degrees Celsius. Auto-anti-I, when we get to that one, we'll talk about I. It's the most common cold agglutinin we're going to see. Anybody, though, that's going to react at room temperature or lower is going to be considered cold agglutinin, and most of the time, this is going to be your IgMs. They like it cold. Warm antibody, anything whose optimum temperature is close to body temperature. These are going to be predominantly your IgGs, and oftentimes these are going to be pretty clinically significant. So stages of antigen-antibody reaction. I got sensitization versus agglutination. So remember I talked your IgGs, small. I'm going to hang on to it. I'm going to attach to it and hang on to it, but I can't do anything else because this arm can't reach a second cell. Versus you got these big IgMs that are like, oh, I'm going to cause major agglutination here. I'm going to grab onto all these cells, and then you can see me. So sensitization is the coating of cells with the antibody. This could happen in vivo or in vitro. cells may hemolyze in vivo. That means in the bloodstream. If I have antibodies floating around, I'm sensitizing my cells, they could start hemolyzing in me. They may hemolyze in vitro, but more commonly, you're going to get them to agglutinate in vivo by using that cross-reacting link we talked about, that we'll talk again later. We'll end up finding something that'll attach to the back end here in the FC portion of our antibodies and agglutinate there. Not visible macroscopically or microscopically. You have to trust me that immunoglobulins exist. We can't see them. You just have to know they exist. Agglutination, though, we're looking at visible clumping. So see here how this IgG has 3 cells and this one has these 4 cells? And so now I've got a little clump of 5 cells. That's agglutination. This only happens in vitro. We don't get agglutination in vivo. Thank goodness. Because you get a clump like that and now you're occluding one of your capillaries. Remember, capillaries are so fine, only 1 cell at a time can go through them. So that would be problematic if that happened in the body. Caused by coated cells being close enough together for antibodies to react with antigens on more than 1 cell. Because of their size, IgMs are really, really good at creating agglutination. So factors affecting, that's a hard word, factors affecting sensitization, specificity, the spatial and chemical fit. Okay. Think if I'm building something with Legos, if I'm going to put pieces together, they have to fit together, right? They have to have the same size opening and the same size pieces to go in, or there's be gaps, or they're not going to fit. That's what we're looking at. Charge affects the shape of the antibody molecule. The pH is going to affect the charge. Temperature, use room temperature or lower for IgM, 37 or higher for IgG. Time, you need to incubate them long enough to ensure maximum interaction, but you don't want to go so long or eventually they'll start falling off their antigens. And then accessibility, making sure nothing is in the way and that the antibodies can reach their antigen. So we're going to talk about these individually here. Specificity. This is the antigen, this is the antibody combined site. This is a nice, good, tight fit. This guy, this is the antigen, this is the antibody, this is a poor fit. You want the shape of the antibody to fit into the shape of the antigen, or the antigen permanent. We want the shape gets developed at the time of immune response. So the first time you get that exposure, your body's going to say what the antigen is going to look like. That's going to fit that antigen. Your immune system is going to say what the antibody is going to look like. It's going to fit that particular shape of antigen on the red cell. Not much we can do in the lab to change specificity. The antigens, antibodies are either matching or they don't. Charge and pH. Red cell surfaces all have this negative charge around them. The antibodies have a positive charge at a slightly acidic pH. So all of our reagents are going to be slightly acidic. Differences in charges attract antibodies to the red cell antigens, and because of that, as I said, blood-based reagents are usually slightly acidic to actually enhance that reaction. So you can see here that it's slightly basic, it's going to repel it. This one is slightly acidic, it's going to help it. Here's your temperature. You can see 37 by 15, where am I at here? I have to get in front of it. 20 minutes. At 15, we're at the maximum reactivity. If working with IgMs, we always want to work at room temperature or colder. If working at IgGs, we want to incubate at 37. So this is an IgM, or I'm sorry, this is an IgG. 37 is going to work best. If I'm incubating at 35, it's going to take longer till I have the maximum agglutination, and 32 is going to take even longer. So we want to make sure to always incubate at whatever it says in the manual. Sometimes we incubate in the refrigerator. Sometimes we incubate at room temperature, just on the counter. Sometimes we actually are doing it in our hot bath. So it's always important to pay attention to your manuals. Time. We want to make sure to give it enough time where we're at this fully maximal antibody uptake, but as we wait longer and longer, that's going to fall back apart. So most of our procedures say incubate 15 minutes up to 30. I always say that's the time to go get your break, go to the bathroom, get a drink, get a bite to eat, because you have that little bit of leeway there. But once you're over 30 minutes, it's going to start falling off and you're going to have to redo it. Accessibility may be effective for antigens that don't stick out very far. If the antigens are like going through the cell wall and just barely kind of peek through that cell wall, we may have a problem getting to it because there's all sorts of water. So you can see these little circles are water molecules. We use PEG in our labs, and PEG is polyethylene glycol, and what it is antifreeze. Works just like antifreeze. And so what's going to happen is when it goes farther, goes like this, it's going to take all those water molecules and it has a higher affinity to the water than the red cells do. And so it'll pull those red cell molecules away from the cell so that our antibodies can get in there and get to those antigens on the red cell walls. Something else we can do for accessibility is we can use enzymes that will help destroy the outer side of the red cell membrane, which will also, if we have something going through the membrane, it'll help that stick out. We'll talk more about that when I get to the enzymes a little bit later. So, factors affecting agglutination. Similar to the factors affecting sensitization, number of antigen sites. The more the sites, the greater the chance for agglutination. Structure of the antibody, polymers like IgM with more antigen binding sites. The size of the antibody, the bigger the antibody, the greater chance of agglutination because it can reach more antigens. The distance between the cells, the closer the cells are, the more easier they are to agglutinate. And then the antigen-antibody ratio. Optimum is 80 parts antibody to 1 part antigen. That's going to be your optimal ratio. Number of sites on the red cell, again, not much we can do about that in the lab. Okay? Enzymes can open them up some more, but some antigens get destroyed by enzymes. It's not a simple routine technique. Oftentimes it's something that's going to be sent to our reference lab for. Size of the structure, IgM, bigger, more likely to break that zeta potential, bridge the gap, and cause agglutination. There's that zeta potential. We can change it. We have this outer layer of cells. These cells actually repel each other. So there's things you can do to reduce the zeta potential. You can put different reagents in it to help those cells go closer together. So here is, kind of shows an IgM. Those cells are kind of repelled. Here's an IgG, and you can see they're so close together, little T-Rexes, you can't grab that other cell, versus an IgM like, oh, hold my beer, right? I'm going to hold all of these things. So you can overcome part of the zeta potential with your IgG with that Coombs antibody. We talked a little bit about it. We'll talk about it multiple times. But what the Coombs antibody is, it's an anti-IgG. And what it does is when you're drawing out the structure of an antibody, you got your antibody binding sites on this end, and you got your FC region on this end. It's a constant region. So what this is an antibody to that constant region. So it's to all human IgG. So it's an anti-IgG. So we put these together and incubate them, we wash them, and then we add that anti-IgG, and now we're able to help bridge that gap a little bit more. Antigen to antibody ratio. We really want to make sure that there's not too much antibody. That's going to form a prozone. You want that 80 to 1. That's going to be your zone, optimum zone of antigen and antibody together to get the best reactions. You can see here in the antigen excess, the prozone, how there's so much antigen that it doesn't ever actually cross-link. It just binds itself to, there's so much of it, it's just all there. Then you got your post zone at this end, and your post zone you have antigen excess, so there's just not enough antibody to cross-link because there's so much antigen, it's all, there's just lots of extras all over the place. We get this optimum 80 to 1 by diluting down our red cell antigens to a 2 to 5. We use 3% cell suspension. So we want that 3% suspension in order to get the optimum 80 to 1 antibody antigen ratio. Reading and grading reactions, agglutination reactions need to be read based on how strong they're holding together. When we grade them, it gives us an indication of how strong and how much antibody is actually there. Some reactions can be done on a slide. Most of them we're going to do in a test tube and we grade them. On a slide, it's positive, negative, because we can't grade them on a slide. However, we put them on a tube, we can. So when we're doing this, we put cells and serum mixed together in a test tube. We centrifuge the tube. By centrifuging the tube, once we put these antigens in serum, the antibodies and antigens in the tube, by spinning them, by centrifuging them, what we're doing is we're pushing those cells together to help them react. That's why we need to make sure before we actually do any readings, we spin it to read them because we want to push them together to help them react. If I just let them sit, eventually you're going to have some agglutination, but not enough. We want them shoved together. Cell button then, we resuspend it over a lighted agglutination, and we observe for what we're looking at for degree of agglutination. So here is a negative tube. When I demoed this in the lab this week, and you can watch the demos online, we shake it. It's called the coffee shakes. We are barely shaking them. We don't want to shake them like this because we're going to break them apart. You just shake your tube gently, rock it, shake it, rock it. And you're wanting to watch how that button comes apart and how the cells spread out. So a negative reaction gets reported as a 0 with a line through it. All the cells swirl off the button as it's gently shaken so that you don't have any chunks. All the cells just individually swirl off. Looks like little wisps of smoke coming off the cell button. And it indicates that the antigen or the antibody that you are testing for is not present when it's negative. The other end of things is the 4+. The 4+ is the strongest agglutination possible. This is what we see when we have, for example, when I put anti-A in with my A cells, most of the time I'm going to see this 4+ reaction. All cells are caught up in one clump of agglutination. Cells do not come off the cell button when this tube is gently shaken. I can still take this and whip it and get it to break apart, but that's not telling me anything. I'm going to do the gentle shake, and I want to look for this 4+. Once in a while, you might see a few tiny little satellite clumps around a large clump, but for the most part, you see this one big clump, it's going to be a 4+. We do have at everybody's bench a picture of -1+, 2+, 3+, and 4+, and so you'll have these to look at when you're reading reactions. I also want to note that in your lab manual, we describe these all, and you will always have your lab manual. All exams, all labs, make sure you have it, make sure you've read it before you come in. Make sure you know what you're going to be doing in lab, because that'll make or break you. If you come in a lab and you ask me questions and you don't even have your lab manual open, I'm going to walk away. You need to be following your lab manual. If you're in your lab manual, like, okay, now I'm really confused, I will help you. I'll bend over backwards. But if you want me to come help you and you're not even looking in your lab manual, that's problematic. Because just like in your analysis for all exams, you get to use your lab manual. I want you to learn how to use your lab manual. 3+, the large clump is going to break up into several large clusters, but see how the background's still pretty clear, no unagglutinated cells? This indicates a fairly large amount of antigen, but it's not a full 4+. You got some larger ones, it's a 3. Now we're down to a 2+. You got lots of medium-sized clusters, but your background is still pretty clear. Indicates a moderate amount of antigen or antibody. 1+, the cell button is broken up into numerous small but easily visible clusters, and your background is cloudy. You can't see it really well here, but when you're looking at them, you'll see your background is cloudy with all the individual cells with a couple of tiny little specks of darker cell in it. Weak+, we're really not going to use this much, but what this means is you got a couple tiny clusters that are best observed only when you see the agglutination viewer, and it indicates a very, very small amount. Pretty much when we stop at 1+, we don't take it to the weak. When we look at things microscopically, there's a couple tests where we'll take it to the microscope, and what we're looking for is see these little clusters of cells amongst the sea of individual cells. Those are ones you're not going to see with the light agglutination viewer, and you'll take it to the microscope. We only do this for DATs and HDNs. Mixed field. Mixed field, you only report involving patient cells, and it should only be reported if that patient has received somebody else's blood recently. Because what that means, I've got 2 cell populations or more, if somebody had multiple transfusions in my system. And so I've got these cell populations in my system, and so some of them are going to react to the antibodies, and some of them may not. So, for example, I get a bunch of O blood. Let's say I get a massive transfusion, I get a whole bunch of O blood, and I'm A. When I put the anti-A, it's only going to react to some of the blood. So I might have a nice big strong 4+ or 3+, but there's going to be a lot of cells in the background. And what that means is I got 2 completely different cell populations floating around. Hemolysis is a very significant report. When you see hemolysis in a test tube, you're going to report it out as HEM+. So the supernatant is red or pink. You want to make sure always check that and compare it to what it was when you put it in the centrifuge. If I put clear serum in the centrifuge and now it's pink, that's a problem. Sometimes you end up losing all your cells. Like you go to look like, where'd my button go? My button's gone, it all hemolyzed. That's what's going on. This is a lighted agglutination viewer. The FDA requires that you use a lighted, mirrored, magnifying agglutination viewer whenever you're doing any reactions. When you're doing these in lab, I will show you this week how to do them, but what you're going to do is you're going to pull that agglutination viewer right to the edge of your counter. It is no good to be magnified if I'm looking back here, right? You're going to pull it here, and you're going to be looking in that mirror at your tube. You're not going to be looking at the tube, because the cells are going to be on the bottom in that mirror. You're going to be watching your mirror to see what's going on in that tube, because the mirror magnifies it, and it's lit up where you can actually see all these individual cells. Resuspension technique. We only read one tube at a time. You hold the tube upright over the mirror. You're going to check for supernatant for hemolysis, and you're gently going to shake the tube to resuspend, and then you're going to tilt it. Shake it and tilt it while watching the mirror as you're doing this to see how those cells are coming off. You want to make sure, are they swirling off and then it looks negative? Great. That's a negative. If they kind of chip off a little bit but then kind of look negative, that's probably going to be a positive. They're chipping off the bottom. So we're really paying attention to how those cells are coming off the bottom and what it looks like. It's important to continue shaking and observing until the entire cell button is suspended. So you can't just stop once a few cells come off. The whole thing has to come off. Read one tube, write the answer down. Read the next tube, write the answer down. Your brain will play tricks with you. If you read them all and think you remember, it's going to put down what you think it's going to be and what it should be, and you miss mistakes. So blood bankers really know how to shake it.